Welcome everybody to the Tuesday, October 12th session of the STEM Education Institute seminar. We are delighted to have three people with us today, and I will introduce them in turn. The title of today's session is A Cure for Undergraduate Research Projects, a community supporting faculty to introduce research into their laboratory classes. CURE standing for Course-Based Undergraduate Research Experience. And our three presenters will tell us their experience with two different courses. Our first presenter is Dr. Amy Springer from the Department of Biochemistry and Molecular Biology. Amy got her bachelor's degree down the road from Mount Holyoke College and then went on to get a PhD at Princeton University, advised by Dr. Molly Schmidt. She then went on to two postdocs, one at Caltech and one at the University of Washington in Seattle, and then, unusually for many in academia, two industry positions at MDS Pan Labs, ProLinks Incorporated, and the Seattle Biomedical Research Institute, which is now called the Center for Infectious Diseases Research. She has been in the Valley prior to coming to UMass. She taught at Mount Holyoke and Amherst colleges and then went on to teach at Siena College and joined UMass in January of 2016. In addition to being a lecturer, she is also the chief undergraduate advisor of the Department of Biochemistry and Molecular Biology and has pioneered this new course-based undergraduate research experience that she will tell us about today. So welcome, Amy. Dr. Jessica Rocheleau is a lecturer in the Department of Biology at UMass. Jessica got her PhD at the UMass Medical School in Worcester and then did a postdoc here at UMass Amherst in the microbiology lab of Dr. Derek Lovely. After which she went on to a faculty position at Western New England University, where she was associate professor, before returning to UMass in fall 2019. She joined to revamp the introductory biology labs, and she has a great interest in finding ways to make biology research accessible to all. Jessica's work in the introductory biology labs CURE program is part of something called CPhages, which stands for the Science Education Alliance Phage Hunters Advancing Genomics and Evolutionary Science program. The intro biology sequence serves 1,200 students every year, and she has gradually been trying to convert all of the introductory labs to be based on a research-based format, and this is what she will tell us about today. Jessica will be joined in her presentation by Madison Riffe, who is a graduate student in the Neuroscience and Behavior program at UMass. Prior to coming to UMass, Madison did her undergraduate degree in biology at West Virginia University. Here at UMass, she works in the lab of Dr. Jerry Downs, using zebrafish as a model for a rare seizure disorder. And she has also been involved in teaching the introductory biology labs. So welcome, Jessica and Madison. And to begin with, we'll start with Amy's presentation.
All right, great. Well, thank you, Shubha. Thank you for the invitation to talk. And so what I want to talk about CURE, and the way I use CURE is to mean course-based undergraduate research experiences. I want to first give a little introduction about why we want to use CURE, why we want to do undergraduate research in course-based format, and a little bit about how the program that I'm involved in was just a community supporting faculty teaching that. And then we'll hopefully segue into what Jessica's going to talk about afterwards. So, just click in the main window. Yeah. We thought we practiced this part. Okay. So, what is undergraduate research? So, For undergraduates to have an opportunity to experience the inquiry-based, scientific thinking-based practice of doing science rather than just stepping through procedures. And there's lots of evidence that there are benefits to undergraduates to having these experiences, and particularly in enhancing their student learning and their retention in the major. We see better enrollment in graduate programs and career longevity in STEM, critical thinking, problem solving skills, and even just that understanding of research methodology coming out of these experiences. There are several references, and I have some at the end of my slideshow, but that students participating in research, a lot of the data is attitudinal, but they report gains in independence, intrinsic motivation to learn, active participation in courses, and so forth. You think you want me to have a microphone on? I'm wondering.
Except there's no battery.
No, there's no battery, so I'm going to just talk loudly through the mask. Of course, there are challenges, and particularly in an institution like this, there are challenges to giving every undergraduate that research experience. So if we look at my department, we have about 18 faculty who are active, but not all of them are necessarily conducting research, and although all of them that are taking undergraduates, but we have about 700 total majors, 160 or 180 graduating a year, and we definitely don't have space in our faculty lab for 10 undergraduate students a year. So it's hard for them to find a lab, and it's hard for to find the resources to be able to find for research projects for all these students. Furthermore, faculty are having to teach courses, and if that's a separate thing from their research, that's that's an additional time component for them. So if we can make the research part of their teaching load, that actually helps reduce some of the workload that students have. So I'm going to be focusing really on how we're trying to do this with B&B majors. When Jess gets up here and talks about the entire CNS college, you'll see that she has a different scale problem to worry about. So laboratory classes. And so to make the distinction between what do we think of as a laboratory class and what is a cure, and often, even if you do an inquiry-based project in a teaching lab, often there's a known answer, and the instructor knows the answer. If you do a cure, there is an unknown experimental outcome, and actually there's a hypothesis development component where the student has to come up with a hypothesis for the experiment, to do the experiment and what they think the outcome might be. You get a lot of guidance by faculty and instructors in an inquiry-based setting, but often the cure ends up being much more self-guided and peer-to-peer guidance within their teams are helping each other kind of get through the steps. So the problem really is, how do we provide research projects at scale? And particularly for us, we want students doing wet lab research. We want them with their hands, we want them using pipettes, we want them working with proteins and genes. And so it's a bit of a challenge to think of how to scale up to that size. And this is where, for me, I was attracted to the idea of this protein-based cure around malate dehydrogenase. And this started as a two-semester, two-step cure sequence by Dr. Joe Provost and Ellis Bell, two of my colleagues working with the MCC. And they had this great model, which was basically a year-long research model. But they thought it had enough legs in it that we could expand it to work in more different environments. So first of all, why did they pick malate dehydrogenase? What do we like about it? So it's a pretty well-known enzyme. There's a very simple assay that you can do on the benchtop. Very basic function. This is actually an enzyme that's a part of the citric acid or Krebs cycle, which probably most people have heard about. Very highly conserved. So there's at least one MDH in every organism, often two. The structure is known for many of the different isozymes. And yet there is a significant amount of diversity as you go across the taxa. So there's a lot of questions you can ask about comparing and contrasting enzymes from different organelles or from different organisms. And while we know a lot about it at a basic level, there's still a lot left to be studied. Why is it an important enzyme? So I mentioned the Krebs cycle. This is one of the important steps in pathways in intermediate metabolism. And so malate dehydrogenase is one of the important, it can be an important regulatory step in determining whether something stays in the Krebs cycle or moves on to biosynthetic pathways. It's also involved in the process of gluconeogenesis and in the movement of electrons for redox valves, which is a system called the malate splenic shuttle. So it turns out it's an important enzyme in the juncture of several different pathways. So in addition to it just being an easy enzyme to work with, it has a lot of potential interesting impacts on cells and organisms. There are also many different ways we can interrogate and ask questions about it. We can look at it at the structure and function level. We can look at it in terms of catalysis. What are the enzyme kinetics? How does an enzyme work? Can we change its substrate range and so forth? And we can look at it at the regulation level and look at modifications and so forth. So there's several different kinds of questions. Everybody can be working on malate dehydrogenase, but there's no end to the number of questions we can look at. We can look at questions that are better applicable to lower division courses, like a Gen Chem or an intermediate biochemistry course, or upper level courses. And we can really align the kinds of experiments we do with the foundational concepts, in particular the foundational concepts in our society, the Society for Biochemistry and Molecular Biology. So starting with Dr. Provost and Bell, we have been building this, what we call the MDH Cure Community, or the MCC, we just finished a 3-year IUS grant and are just starting an NSF RCN UBE that just started this month. We're creating basically a network of faculty at various different institutions that are doing malate dehydrogenase based projects in their teaching laboratories. And it's given us an opportunity to get faculty together and talk about sharing their experiences, their troubleshooting, and really get some good opportunities to have students focus projects in their courses. We also really want to keep making this more possible to more faculty. We're finding lots and lots of faculty who are interested in doing this, including at under-resourced institutions and community colleges. So the goal of our latest NSF RCN UBE grant is to make the resources and networking more available and lower barriers for new faculty who want to adopt cures in their own courses. So we are 26 institutions and counting. Last time I looked, these are some of the examples of some of the people that are in it, and the website here, which I can share with people, has a lot of different information about the kinds of resources we have, the kinds of projects people are doing, and so forth. So just to give you an example, I will talk about my angle where I look at malate dehydrogenase and how I use the resources from the MCC in order to do that. So my background is in microbes and most recently in working on trypanosomes, which are a set of parasitic unicellular eukaryotes. They're protozoan parasites that all of which that we know of are parasitic. And they have some very interesting, I love this picture because it gives you some sense of the different morphologies you get. The one that I study mostly is called Trypanosoma brucei, and it is the causative agent for African sleeping sickness. This is a disease that impacts the sub-Saharan Africa. And it is the disease that is transmitted by the tsetse fly. So if you do the New York Times crossword puzzle, there's always this like menace in Africa, and that's always tsetse fly. That's where most people hear about it. But this is the disease that it transmits, and it's estimated 40 to 50,000 deaths a year. It's actually very difficult to really get good data on the deaths because some of them go unreported. But there are some 60 million people living in affected regions. And it's not just a human disease, it's actually also a disease of livestock. And so it impacts not only human health, but also their potential, food sources and economic livelihood. So in order to come up with good treatments for a pathogen like this, it's really important to understand the basic biology. Especially when you're dealing with a eukaryotic parasite, you don't want to target something that's also going to target human functional proteins. So you want to look at things that have some differences in our immune. So why malate dehydrogenase in C. brucei? Well, it turns out this one has not one, not two, but three different malate dehydrogenases in it. The mitochondrial and the cytosolic are what you find in a lot of different organisms, but it also has this third organelle called the glycosome, which is actually a membrane in which many of the steps of glycolysis occur. And that has its own MDH in it. So I told you that these enzymes are very important in regulating kind of different pathway checkpoints in various pathways. So this one clearly has an additional level of that. So we're really interested in finding out kind of more about this enzyme and see if we can predict what its role is in the cell. We can look at some comparisons between them, see how they differ from one another, how they differ from their orthologs in other organisms, in particular how they differ from humans. So where we started out is actually looking for interactions between MDH and other enzymes. There's a lot of data to suggest that it interacts with its immediate neighbors in the citric acid cyclic pathway. And that's important for its role in humans, for example. These organisms use the citric acid cycle or Krebs cycle very differently, and so it's quite possible that they have different or no interaction. And so that's one of the things our students have been interested in. There are also enzymes that the cytosolic version of MDH are likely to be interacting with. And so we've been going through the process of trying to figure out if these enzymes are forming interactions. The model is that they form interactions to allow the substrate to channel much more efficiently from one step to the next. And so we can learn a lot about kind of how they're using their enzymes if we can look into these interactions. So when students come into my class, and this is an upper level class, so these are generally seniors in biochemistry, and they've been doing a lot of coursework. They have a lot of material under their belt, but these are actually students who are not doing independent research on their own. So those students don't take this class. And so when they come in, we spend about a month kind of getting them up to speed on some of the research techniques, but also getting into the literature, talking about what we know about MDH and what we know about contaminants, and asking them to come up with a hypothesis. And to be honest, I give them a lot of strong suggestions because we have things that someone has been working on and it needs someone to take the next step, or this is something we'd like to get started. But one of the things we can do, for example, is align some of the isoforms and look for things that are similar, say with the human, or similar between the mitochondrial and the glycosomal, and we can make decisions about, do we want to mutate some region, or do we want to swap a region from one isosome to the other and see if it makes it behave more like the original. So these are the kinds of things my students are doing, and they can come up with a hypothesis, and we set up to do the experiment, and then they give a presentation on the experiment. The hypothesis building portion is really important to the cure. It's really important to getting them invested in it and really thinking about how you come up with something you don't know what the outcome is. You may have a good prediction or you may not have a good prediction for what the outcome is, but you have some expectation. And so this is where the MCC intersects with my course. I mean, I've been working with them on protocols and resources and so forth, but we do a Zoom call just after the students have finished their hypothesis. And they get to talk to an expert who in fact studies MDH and citrate synthase, MDH interactions with other proteins. And they talk a little bit about their projects, and he can share a little bit of what he knows with his or what his lab is doing. And I find this is a really engaging part for the students because they realize that there's someone not at UMass that's interested in this, someone that is, from elsewhere, and there's a community out there. So the community is not as helpful to me. It is very helpful to me for troubleshooting and problem solving and, oh my God, what am I going to do next? But it's really helpful for the students to get them really engaged that what they're doing somebody else cares about. So just going to summarize kind of what's been going on, but these are some of the things we've done in the last few years. We've been cloning, we've been optimizing expression, we've cloned some MDHs from some related trypanosomes, we've cloned a bunch of genes that we think might be adjacent pathways so that we can start doing interactions. We need to start optimizing the interaction assay. We've got a couple different ways to do it, but one of them that's really interesting is a thermal melt that changes when there's an interaction. We've also, students who design mutants, specific mutants, including we're really interested in a set of mutants that look like they're modified in a certain way that may correlate with interaction with the citrate synthase enzyme. And they also are kind of looking at other regions that look like they might be conserved and involved in this kind of interaction. So that's the sort of projects my students have been doing. So is it working? So there's now an increasing body of literature of reporting about how students are responding to cures and what they think of them. And we definitely see over and over again that it really has a significant effect on their sense of ownership in their projects and their commitment to STEM and their career intentions in STEM. Our own grant, the IH that just finished, we found that a definite increased interest in pursuing subsequent science. We were fortunate in our community to be able to bring some community colleges into it. Because we're a big enough community, we could be working with them. Better research labs could sometimes help them with ideas or try to do experiments. And it was in these community colleges where we saw the most dramatic increases in this intentional in STEM. So we think that it has a bigger impact the earlier you are, and maybe the less exposure you have to research or graduate your institution. We also compared the difference between having a full-length cure like mine, where the whole semester is a cure, versus squeezing it into part of your semester. And both of them actually had a positive impact on students' attitude or attention. The longer ones were better, but some people don't have, especially if you're trying to squeeze it into a Gen Chem course or something, you don't have the luxury of giving up your entire semester. So it did seem like there were ways to do that as well. And then lastly, I got some quotes from some of my students. And I'm always hearing back, especially these students are seniors. And so it's often their last semester or next to last semester. And I'm often hearing back from them that they went off and got a job in a lab because of this research experience. This was enough research experience for them to get a job in a lab. I love this one. I was pushing out boundaries for myself, particularly related to my graduate coursework. It's an undergraduate. I also learned how to celebrate small victories. The experience I got with lab procedures, documentation, fundamentals has been invaluable. And then I knew I love this one. I knew I wanted to work in research, but I didn't have much experience working in a lab. I was nervous that I have a hard time getting a job, but she got a job right not long after that. So we think it's working. Unfortunately, I haven't seen any studies that look at students 2-3 years out, which is really love to see. But it's certainly a lot more fun to work with them and see them go through this experience. So I think I'm gonna stop and let You're gonna switch over the slides.
I will switch over the sides. Meanwhile, if anybody has a question for
Question for
Amy
me now?
while we are doing the changeover, please
Oh, yeah.
go ahead
Yeah.
and ask. You want to
I'll
stand in
stand
front of
up.
that screen and see if
Stand over
anyone
there,
else.
Megan.
We're doing this in a multimedia way. Is there anything on the chat? Nothing on the chat.
So I got a question.
Yes, there's a question from the floor. Excellent.
Do you find a challenge with student-to-student transmission of ideas? Not as in the research side, but as in institutional knowledge contaminating next semester's effort to start. Like they talk to their fellow student, well, I did this. Okay, I'm going to do the same thing.
So do students, students have a lot of input on each other.
Yeah.
So for because I teach it once a year, they've graduated before the next round comes, so they're not about the same projects, they're not doing that, but they certainly could talk to students in the semester before who were working on a different project with a different professor and might have done different things. That's not necessarily a bad thing. They might say, my friend said they did it this way, what do you think? And then we can have that conversation, and it might be the right thing or the wrong thing, but I do encourage them to come up with ideas, and I try not to give away that it's the wrong thing too early if it is. I try to let them get to that. But that's a good question. Thank you.
That for me, I love this idea. I'm finding in physics because we're more introductory stuff.
Yeah.
From year to year, we get contaminated from.
Right. Well, and you're
they
probably
lose
doing
the opportunity to think for themselves.
right. And you're probably doing the same thing. So that's of course
Yeah, I don't
the
have
beauty of
this. But
that's the beauty of not doing the same thing. And unknown outcome is that might have worked, but were they doing a different project?
yeah.
Amy, you have a couple of questions
All right.
from the chat. Drew Stevens asked us, what does full-length cure mean? Don't you need to teach the base techniques first?
So
When do they do their hypothesis?
yeah. That is such a great question. Yeah. So full length means we start thinking about the project from the beginning, but it's true. I generally get the hypothesis out of them by about 4 or 5 weeks. So we spend a lot of time reading and doing that in the beginning. I also spend the rest of the semester refining the hypothesis because I think they understand it better at the end than at the beginning. But yes, we have the first 4 weeks we're doing some basic techniques, but they're related and they're actually with an MDH, not with an MDH, so that they're learning the techniques that they're about to use.
Go.
It's good. It just now went off. Oh, can't see. Oh, okay.
You can see it. Yeah, go ahead. Sorry, Amy.
Good.
There's one more
Any other
question. Yes, are there ways for them to reproduce already established research before they dig into doing new work? This is from Sam Krishnamurthy.
So yeah, it's a good question. I mean, like I said, we romped through some basic techniques at the beginning, which actually isn't used to because they need to refine their technique before we want to do stuff. In principle, you could have someone, and it might not be a bad idea to have a team do something that a team another year had a result they had found and confirm that they found it. I try not to do that. I mean, try to not to do that so that they think there is a result out there that they should have, right? To make it clear that if they come up with that independently, I'll let them do it again. And if they both confirm it, then that's great. And maybe we publish it. And the goal, of course, is to make this publishable. But I really want them to have that experience of original research. And we can certainly follow up something that we want to publish. We can certainly follow up and repeat it, in the summer or something, but not having to take away someone's original research opportunity by repeating someone else's. Great.
Okay.
Okay.
Thank you, Amy.
Yes, thank you.
On to Jess.
Okay, so I'm going to talk about, should I move this? Just realizing, sorry, for everybody here. That's as far as I can get. I'm going to talk about the C Phages project, which is the full semester cure that we're introducing into the introductory biology labs, which serve many different departments, all of our life science majors. Oh, there we go. Okay. So I'm giving a quick definition here too that I think is very similar to what Amy said, right? So These are projects that engage whole classes of students in addressing a research question or problem, unknown outcome, and something that's of interest to the scientific community, the broader scientific community. So that's what we're doing here as well. And this comes as a response to national calls for engaging more STEM undergraduates in research. So I'm going to use this framework from Aaron Dolan and a little bit of Graham Hackle, just about what effective cures do, and talk to you about how the CPhages project accomplishes these goals. So, technically and conceptually simple, I wouldn't necessarily call what we're doing simple, but it's simple enough. Compatible with flexible scheduling is something that's really important for the intro labs that we've been playing with a lot. Specific milestones that we can hit, structured so students work in parallel, checks for data quality, a repository for sharing this data, assessments that resemble what scientists really do, and then this really important aspect of project ownership. So CPhages stands, I think sometimes folks get confused and they think that there's some marine elements to this, and there's not. The SEA is the Science Education Alliance branch of HHMI. So this is a Howard Hughes Medical Institute sponsored program. And then Phages comes from this, it was a Phage Hunters program that was established by Graham Hackle at the University of Pittsburgh. 12 or 14 years ago. It's an old, it's a well-established program. All of these data on this slide come from, I think, the 10-year mark, so we're well past this, but there are many institutions involved. Most of the students are first-year students that participate in this program. We're at over 100 colleges and universities that participate in this program, so lots of support for faculty who are implementing this. And lots of data now. We're at this, so this was 13,000 isolated phages a couple of years ago. We're now at 18,000 isolated phages, and I think 3,000 or more are completely, their genomes are known and annotated. So it's exciting to be part of this big project. And so the main goal is to find and characterize novel bacteriophages. So these are viruses that infect bacteria. And understanding the diversity, much like Amy was saying, understanding the basic science and diversity inherent in this massive population leads to translational applications. And there's a really exciting one that I'm not going to go into, but phages that were discovered in the CPhages project have been used successfully in phage therapy, in bacteriophage therapy, with an antibiotic-resistant infection. So there's a Nature Medicine article that we always start the semester with. It's like this huge hook, and everyone's like, whoa, wow, this is, we have this clinical element that's exciting. So it's ironic that I'm talking about how technically and conceptually simple this is in a week when all of our stuff got contaminated. So theoretically, this is technically and conceptually simple. So we have these viruses that infect bacteria. In this project, there are several different genus and species of bacteria that we could choose. So that lets us sort of branch into different areas if we want to. And we go digging in the soil, often around UMass, and mix our filtered soil sample with our bacterial host of choice. And if we have found bacteriophages that infect our host bacteria, we will see a plate that looks like this with plaques. So these are clearings where the phage has killed and exploded the bacterial cells in that location. Right, so it's an easy assay, an old assay where you can see, are there phages in my sample? And then we use that over and over again to purify the phages that we find, to make large quantities of them, and then we get to characterize them. We can take advantage of the fact that the global phage population is massive. And so the chances are that when we dig in the soil around UMass or in front of the Bank of America, where we found our most exciting phage yet, we're going to find novel phages that are different from any phages that we've found before. So that part is exciting. The population is old, dynamic, genetically diverse, so there's lots of exciting and rich information to find. And so these are the main experimental questions that we're asking in this program. It's just like, how are they related to each other? How do they get to be that way? And then again, sometimes these awesome translational applications emerge from this collection of data. We have a limited, especially given the hosts that we're using, sort of a limited scope of sizes and shapes that we expect to see. And part of our first semester discovery project involves seeing our bacteriophages using electron microscopy, which is a really exciting part. We're doing that this week, actually. Today, we just mounted our samples on grids. So students get to see what their phage actually looks like, and that's a big part of the project ownership piece, too. So we have these multiple milestones. It really is a 2-semester project, but our bio lab is a 1-semester experience in the first year. And so our goal is to have all of the students who take the introductory biology lab to do this phage discovery project where they're finding the phages, purifying them, and then doing some basic characterization. And they end by extracting DNA that we then can send on to the follow-up course that right now is a upper level elective is where it's sitting. And so in that course, we start with the sequence, the genome sequence of 1 or more phages that our students have found here at UMass. And nobody's ever laid eyes on this sequence before. And then we find the genes that are there and annotate, where do these genes, where do they start? What do they do? And do some comparative genomics to see how do these bacteriophages genomes compare to what we already know and what's already in our database. We have checks for data quality and repositories for sharing data. So I sort of within our program, a big focus is keeping lab notebooks and learning how, developing and mastering that skill. We have undergraduate and graduate TAs who help leave comments and give support with keeping our lab notebooks. And then sort of at a program-wide level, there is an actinobacteriophage database where each phage that is discovered has an entry. The students name their phage. There are some elaborate naming rules that involve not naming your phage after Nicholas Cage. It's like the #1 rule. People try it every year. So we get the location, we have our GPS coordinates for every single bacteriophage that's been found, temperatures, all the main features. The electron micrographs go in here, a link to what the plaques look like on a plate, so you can really mine this database and anybody can for information. And
Jess,
then
can I ask you, how do they do step 0, that it's a new phage? How do you do that? Figure out that it's not a phage.
How do we know? Yeah, so we don't know that it's new. We assume that it's new until we know it's not.
Okay.
So sometimes, once we go to sequence the genome, then we really see how sometimes there are small numbers of nucleotide differences. So it's novel, but not that novel. And we've had one at UMass so far that was identical. And so then what they do actually in the database is they will say, this is an identical phage to, and they link it over to the other phage page and say these two are identical. But there's some that we just wouldn't know until we sequence. Yeah, good question. Once we have the sequences and we finish the second semester and we annotate that genome, we find all the genes, we know where they start, that goes as an entry into GenBank and NCBI, which is International Genome Database, and our students are listed as authors on that entry. So that's a really nice sharing data piece and project ownership piece where the students know that their information is being shared with scientists all over the world and used by those folks. Some assessments that resemble the work of scientists. So we do technique checks and lab notebooks, as I discussed, and reports. We've done poster presentations, obviously, this is from pre-COVID, our first semester in 2019. There's an annual C symposium that our students attend with other students from other participating CPAGES schools. And we've had, even during COVID, we've had multiple students present posters at this symposium, which is exciting and an awesome experience. And so, they're really doing the work with scientists as first-year students in this course. And this is, Nora and Sam from our first semester, like so delighted to see their page on the screen. And it was a type that we hadn't ever found before. So, so we have some exciting moments. So here's our other project ownership pieces that they get to name their page. These are all the ones that we have images for so far since 2019. So we'll be adding probably actually 24 more this semester. So we're scaling this up. Here's sort of where we've been and where we're going with this program. So in our intro bio lab, we run 10 sections usually every fall that serves 240 students. Most of the students though, almost sometimes 1,000, close to 1,000, take the course in the spring because the first semester lecture is a prerequisite. So most students take it in the spring. Those who don't get to will take it the following fall. So we started with, a pilot section, we got to 2, COVID hit, and we ended up running in fall 2020, all 10 sections, we did a remote kind of modified genomics course that Madison was a TA for. So she's kind of seen all the sides of this project now. And now, let's see, we did 2 sections last semester, and now we're here in fall 2021 with almost 100 students enrolled. We're hoping by the spring to get, a quarter of our labs doing this program, and then by next year to fully implement it. And then our bioinformatics course right now is serving, 35 students a year although we'd love to have more opportunities for all of the students who are coming out of the intro course to do the second part. Let's see, so we don't have a lot of data ourselves since we're pretty new to this, but because CPAGES is a national program, we have lots of data on the whole. And so there are some, the main assessment that's done for all schools that are enrolled in CPAGES is this KIDS assessment, which measures intention to persist in science and looks at several different categories. So project ownership, content, project ownership emotion, self-efficacy, science identity, science community values, and networking. And so this is a comparison of almost 3,000 students at 67 institutions, and those that were enrolled in CPAGES in blue had higher scores in all of these. So this is their intention, stating their intentions at the end of the course, compared with students who took non-research traditional labs in yellow. We can also see that if we compare the mean number of science courses that the traditional students took over the course following the CPAGES program, we see that CPAGES students overall took more science courses after taking CPAGES than traditional students. And then retention is excellent. So from first to second year, we see much higher retention among students that took CPAGES, and even improved grades in CPAGES students in their accompanying lecture course than those who were in sort of a traditional intro bio course. I have a few student testimonials too. It's funny to see the similar structure. So we see that students overall have an awesome experience. They have a great time. I know some nice revelations. I think they're always just saying like exactly, yes, that's exactly what we wanted you to say, right? Like I always thought scientists knew exactly what they were doing. Like no, we don't. Like I like that you didn't know what the result was going to be. We had something tangible at the end. I was doing something valuable with my time. I feel like I'm contributing to society. So that ownership piece is big. Students see that. They see that this is a way that science works that they haven't encountered before. in other labs where it was just like, I thought you were supposed to know the answer, or it doesn't work or doesn't it work? So this is a really different model, like, oh, it didn't work, we have to repeat it. And they're like, right, oh, I guess we do, you know, so that's an exciting part of this. And then, oh, this is, sorry, I'm still here. Yes. So Madison Riffe is gonna come up, and she's a graduate TA who has, this is her third semester doing C pages. You are the graduate TA who has done the most C pages of all the graduate TAs. So give us your pages.
Yeah, I really wanted to just talk about some of my interactions with the students, how I feel like they benefit in this course. I also have TA the traditional bio lab, the intro bio lab. So seeing the CPA students compared to students that are enrolled in like the traditional intro bio lab, just a few observations. First, I think CPA students become familiar with authentic scientific research, which really is like where you just left off, just appreciating like, we don't know the answer and sitting down together and figuring it out. Then you can like come into the lab and be like, what am I doing today? I'm like, I don't know, did you look at your plates? I haven't looked at them yet. And it's just like a really awesome, I think, experience to like sit down, we're figuring out what we're doing today, every day together. And it's kind of like an authentic troubleshooting experience that they haven't had in other bio labs, or probably any other lab where, like you said, the result is known and we're working to figure out if it works or not. I think they also gain confidence in their lab technique and their troubleshooting abilities across the semester. So they get more and more comfortable with the work, both with the physical techniques, and then it's amazing to watch them gain proficiency in the microbiology techniques, but to also gain proficiency in the troubleshooting and to walk over and they have like looked at their plates for the day. They've come up with like what went wrong, what went well, and they have a plan to move forward and like tackle it all by themselves. And of course, at the beginning, you have to sit with them and help them make that plan. And then I think as they go through the semester, they get more and more autonomous and like making that by themselves. And that's super cool. Let's see, they also build more personal connections with peers and instructors. And I super appreciated seeing like that networking bar on the CPAGES outcomes graphs there for the PITS assessment. So I think something about like sitting down and troubleshooting together and like working through a real project really brings out some of the personal connections that you would get in like a research lab, where the people that are sitting with you are more than just like these randomly assigned people that you're working with for the semester. They really become like almost like lab mates, and that's how it feels as the instructor too. And so I feel like something about like sitting down and doing real research, the conversations that we have, I think are somewhat different. I feel like it's more adjacent to like, you know, they're more likely to ask me, like, is this the kind of research you do in your lab? Like, what do you do all day? And then we talk about like career conversations and just things that I feel like those conversations come up a lot more naturally than they did in biology labs that aren't this sort of research-based thing. They also get so close with their like lab mates, their lab partners. If they're gonna miss class, they'll email me and say, I already reached out to my lab partners. They know I'm gonna miss, they said it's fine. So they really, I think, build connections with each other that are stronger and they seem more like genuine than in a traditional lab. And then as far as myself, as a graduate teaching assistant, I also get a lot of benefits from being part of this. I'm interested in teaching as a career, and so it's super cool to be part of like the behind the scenes action of facilitating the course. And Jess is terrific that she like runs things by me, and there's another graduate TA for the course. And we're part of like decision making, and like I get to feel like this is a product that I am also helping to facilitate and design. And that feels like a really cool, probably one of my best like career development experiences at UMass has been through like being part of facilitating this, which is cool. I also get to share science that I'm excited about. And so I think one of the coolest things about like learning science is learning it from someone that's like so excited to share it with you. And it's, you know, it's much easier to be so excited to share it with them when it's super cool science. And so like, I think the project's cool. I get to be like really jazzed about it and I get to share it out with them. And I think that makes it a better experience for both of us. And last, certainly not least, I get to be part of a supportive team of instructors and staff. And so, yeah, like, I don't know, everyone knows grad school is very hard. The more support you can get, the better. And so, especially on weeks when it's like all of my lab work is like, oh, it's rough. It's so great to come in and be part of like a really supportive team, and to like have, you know, a goal, and we can sit down and like, and show the students a fantastic week of lab work. And the support there of the team, I think, is much more intimate than it would be in a normal lab where you're not part of like behind the scenes work with facilities. Those are all my thoughts. Happy to answer questions. Yeah, for sure.
Oh, thank you, Jess and Madison. If people have questions, you can unmute yourself and ask, or type in the chat. So you began this by saying that it's funny that you're saying this in a week where your samples got contaminated. How do students deal with setbacks? Because in biology,
Yeah.
particularly, there are quite a few, right?
Right, yeah, so it's interesting to see how different students deal differently with setbacks. Like, we've had students, sometimes, especially when we're running in the spring, the first job is to go out and dig soil, and sometimes it's been very cold, and like, you have to chip away at it, and our success rate is a little bit lower than in the fall when things are thawed out. And so sometimes students have had to try, 2 or 3 or 4 times to just isolate a phage, to get plaques on a plate at all. And some groups are like, no, we're gonna keep trying, like, we want our own phage, although we often have extras to start with that I will just collect a bunch of soils just in case. And so we're like, we have one for you, do you want, and some groups are like, yes, we want it, like, we get it, we did this part, and some groups are, no, but we have sort of built in that kind of thing where we don't want any one group to flounder for the entire semester and never get through all of the stages. And so it's a conversation that we have with each group of like, you've done this many times, here are your choices. By next week, we're gonna make you adopt a phage. So you could try one more time. But, in general, I think they understand that the goal is for you to have a phage for your group. And so, we need to keep trying. Yeah, this week was tough because we have something happening, I think, systemically, where everything that was prepped got contaminated, and students had made a lot of plates. And so they were like, but I mean, this is science, this is how it works. We can move on and we were actually ready to prep stuff for microscopy, so we did that today. But I think it helps them appreciate in a way that like there is another side of this, like that we all have to be working together to make this happen. And I think sometimes the students are relieved to find out that it's not them, right? They're like, oh, it wasn't something that I did wrong, it happened to everybody, cool, right? Yeah.
Okay, from Elizabeth, would you compare the resources required for the Cure version of IntroBio compared to traditional offerings?
The resources, in terms of supplies and equipment and money.
I suppose, yes.
Yes. And people, it's definitely more intensive, for sure, and more expensive. Yeah. Than the traditional experience, but I think more rewarding for everybody and a much richer experience.
So does some of the support come from
Yes.
Elizabeth mentions the HHMI?
It's coming from HHMI right now. So we have this 5-year grant that has really made this entirely possible at UMass, right? And I neglected to put that on the last slide. But this is supported by HHMI, and so this has given us all of the equipment that we need to get this rolling, and then we'll be poised to hopefully support it ongoing for everybody.
Can I ask a follow-up?
Yeah.
So one of the challenges we found in physics was, obviously we're not doing this, but we're trying to do equivalent ideas.
Yeah.
Is sustainability.
Yeah.
And we're finding after, how many years into this now? 7 years in, that we are gonna have to pull back because sustainability is an issue.
And can you tell me what's the exact sustainability issue?
People.
That it's just
People willing to put in the labor.
Yeah.
Otherwise, it's you.
Yeah.
Forever, and
Yeah.
And that makes it's fun.
Yeah.
It's awesome, and there's no, but not everybody wants to put the work in.
Right.
It takes, and that becomes a sustainability issue.
Yeah, I mean, we are really fortunate to have Great support, great lab prep support in our department, and so that's a piece that's super helpful and very important. And we also have contributing faculty from other departments who are involved in the H2Y grant and see. So Peter Chen from biochemistry and molecular biology, Sloan Segrist from microbiology, and so that sort of team, network is, I think, going to be helpful for us to continue to sustain this.
Yeah, and I think the community aspect does help with
Yes.
sustainability.
For sure, and I think that the C pages community helps because there's constantly like, here's a new direction you could go, or here's some support for this particular part of this. And I think also, the graduate TA network is very nice because it, you get this new enthusiasm with each new batch of graduate TAs. And that, so it's a double-edged sword. There's a lot of work there, there's a lot of training to do there, but there's also, I think, a lot of reward and energy in that group.
Jennifer Normanley points out there was an article published that attempted to calculate the cost benefits of cures and came to the conclusion that they are worth the investment.
Yeah.
Yeah.
And Elizabeth says also it's worth mentioning that underrepresented populations of students, sorry, I'm just trying to scroll, thrive in the cure environment.
Yes.
Yeah.
Yeah, so I also neglected to say that I put it in one of those slides, the cohort that we first started with is the Biopioneers RAP, which is what is RAP stand for? It's the
Residential
Academic program.
Yeah, which is a group of first-gen underrepresented minority students, and we started with them as a cohort. A lot of them are now our undergraduate TAs. And so that's where sort of the leadership of this program has been, and we're really still focusing on as we scale up, like the first, we're always going to have the Biopioneers in the C pages sections and pull heavily from them. And I think it has been an incredible experience for a lot of those students who semester after semester have been with us as TAs.
Yes.
Yep.
Elizabeth says to you, Heath, that, for Heath, if student outcomes are demonstrated to be improved, we need to make the case for resources to continue the course.
Yeah, no, we
Yeah,
can easily
right.
make that case.
Nice to have the dean on
Well, the reason I asked the question though is for us, we've run into this sustainability barrier after about 6 years.
Yeah, interesting.
There's a certain amount of energy,
And then
it's no longer new.
Right.
And newness, it's exciting for you because it's brand new.
Yeah.
And you run out of, I'm asking the question.
Yeah, that's
also.
it's a good question.
She
Just
did
start
say 10
thinking.
to the 31 pages.
Not to, if you haven't hit that spot yet, but if you,
Yeah.
we didn't think about it early enough,
Yeah.
thinking about sustainability when you still have gas,
What would you have done differently?
that I haven't got the answer to yet.
Yeah.
I don't. And we're not, it really is trying to get more people to do, because as you've discovered, this is a lot more work. And this is work for you than a traditional thing.
Yeah, I wouldn't say it's a lot more work for me, though, in terms of maintenance. Although I have no idea what that looks like yet, because I'm running both. I'm running both the traditional lab and the C page, so that's a lot more work, but I can't really, it will be exciting when we get to the place
When
of
you get rid of
spaces
the old and.
that we can get rid of the old and have the videos recorded and all the curriculum materials written. I mean, I'm still like, we went remote, we went this, we went that.
Yeah, no, you had all. all the
There was
chaos in
so
the
much
middle.
disruption that, to me, like, doing the same thing for a few years sounds.
I think also it's renewing in a way that doing the same lab over and over again isn't.
Yes,
It's a
yes.
fulfilling thing as an instructor.
I think so, yeah, because there's always something different that happens. I mean, the discovery semester a little bit less so than than the genomics semester, the bioinformatics semester, but still, yeah, you're kind of going through a lot of the, similar steps, but with really different outcomes and different issues and different, yeah, we're still finding like, oh, we've never seen this problem before. What do we, that's new. How do we deal with this? Yeah.
So, to Madison's last line, there be part of a supportive team of instructors and staff. Elizabeth asks, does serving as an undergraduate TA in CFAGES have many benefits? So how do you include them as your team?
Yeah, I think everything that I said of the student benefits is even more true for the undergraduate TA. So some of them, I like really stuck with me. Our TA training, one of the undergrad TAs was like, this has been the most impactful experience of my undergrad career. Like some of them are so part of it, they know the science so well, they are like really invested in this entire CFAGES program. It's like, I think it is a huge source of, it seems like a huge source of.
They probably know it more than a lot of the grad students.
Yeah.
Yeah.
Yes, yes.
Yeah.
And yeah, like some of them are still, some of the original lab students that you had, like your pilot semester are now undergraduate TAs, they're like still TA with their lab partner. It seems like a big source of community and like.
Every semester they want to be there.
They do,
Yeah,
they
yeah.
want to be part of it.
Yeah.
Which is, for me, is awesome. I want them to be there too.
Yeah. And you said there's a plan to make all the introductory labs into CFAGES. So have you had any pushback on that, or has there been good buy-in? Because people might say, and this is, I've heard this before, that there are some techniques that you learn in those introductory labs that are left out. But
so, that I considered that too. And when I first started, I gave a talk to the biology department that was, similar to this, a little different. But most of the feedback was, wow, we would love our students to be able to keep a good lab notebook, know how to do these things, troubleshoot, think critically about what they're doing. That seems like it's translatable to whether or not you're working on plants or bacteria or MDH or, so those skills, and I think that you're getting a lot more depth. And the richness that comes with that depth then floating across the surface of many different topics, which is what we do in the traditional intro lab, where it feels quite disjointed sometimes of like, well, now we're doing this, now we're doing that, now we're doing that. And this, you have that continuity and you have the depth. People really get to understand what they're doing and why in a way that they can't in a more traditional type lab.
And the other thing for both of you, I mean, you're doing it at year 1 and you're doing it in year 4. How is that different? What are the different challenges for doing it with freshmen versus seniors?
Yeah, and this is right. We're noticing some new challenges this semester, especially having. I think this is our first time where we really had full labs where we have 24 students. And so it feels chaotic. But like Madison will be like, it's just really chaotic in there. I'm like, yep, this is, it's a chaotic experience. I think having the support of multiple undergrad TAs is necessary.
Yes.
And that really helps to manage it.
Yeah, I guess scaling up is challenging because maybe this earlier cohorts were sort of self-selected to be people who wanted to do this. And now...
right, exactly. I mean, I think there's definitely some excitement. There's excitement and buy-in, but still, all of a sudden, somebody's like going rogue and doing something. You're like, wait, what are you doing? So we have to keep close contact with what's happening with their students, and we need to pay a lot of attention to them. So I think that just starting with those basic techniques, it's a lot. There's a lot to learn.
Yes.
Yeah. Whereas, so right now, because the students I'm teaching in their senior year mostly haven't had a peer experience before that. It's easy because they wanted a peer experience, they wanted a research experience. They also, I can ask them to think deeper and do harder questions than if they were in their first year. And of course then the challenge is how much of that harder question can they get to in a semester, and also, somebody asked a question about having them not repeat the same thing. Well, you kind of want somebody to repeat it to know that it's working and so forth. So there are challenges at kind of that intellectual level that projects are. what it's gonna be interesting to see is as they are now 100% C pages, how's that gonna change our intermediate lab course and our senior lab course? How's it gonna change it? That they've now got this sense of a project and
Yeah.
ownership and, or am I gonna see fewer of them because they're all gonna have found their way into individual labs?
Yeah.
But it's a good problem to have. I'm not worried about it.
I mean, I think it can only benefit.
It can only benefit, yeah.
Multiple, yeah.
So I have a question, two questions, one related to the chaos. Having, no, because I know exactly what you're talking about. And a lot of people struggle with the chaos.
Yeah.
And you gotta let the chaos go. But part of the chaos comes the introductory course because the diversity of student. Not everybody is as prepared. And that's a level of chaos that, you know, is gonna be there forever.
Yeah.
And so I guess the question is how do you deal with it? And then a second totally different question is how could courses like Physics 131 what could we do to help move students along, not in the technical skills associated with the biology, but it's in thinking skills. The mindset is, I think, what we can help with.
Right.
To know that, you know, you don't have to actually have the answer.
Yeah.
And two, it's okay to struggle and be in the chaos and not be a failure mindset.
Right. Right. I mean, I think that some of the chaos, you said it comes from preparation, but I don't think it comes from preparation in terms of like their experiences in high school necessarily, because nobody's isolated a page before.
Yeah.
So maybe some people have held pipettes before and done a little bit of pipette, but I don't know that anybody has that much of a physical advantage. I think the preparation really comes more from like, did you watch the pre-lab video and like read the materials for today? Are you listening to the graduate TA? Then like you're gonna be less chaotic than if you are not paying attention at all and Madison stops talking and then they're like, what are we doing? You know, so, and that's where some of the chaos comes in too. And that's, I don't know, an ever-present struggle, right? To make sure that folks are prepared for when they want.
Yeah, and so but I'm thinking there's two types of chaos I've witnessed in a similar environment. Is the chaos at the team level.
Yeah.
And there's also, I wouldn't call it chaos, but the fact that you have different teams doing different things.
Yes.
And could be at different stages of the process and learning, and which some people I know struggle with, because that's chaotic to them. They're like, well, they're doing this and they're not there yet, or vice versa.
Yes, yeah, we're still learning how best to manage that, because, and it's, you know, right, some instructors, some TAs are going to naturally be like, well, I'm gonna visit each group and talk to each group, or, you know, we've talked about doing even that with our team of undergraduate TAs. Okay, you guys go to this bench, you go to this bench, you go to that bench, we're gonna talk to these people, we're gonna come back. And I think that I have some more thinking to do in terms of the best way to use all of these people. And I think it also though does depend on all of the personalities on that team and how they want to approach it. And I'd say the two TAs we have this semester have very different approaches. And sometimes, but both approaches are very good in different circumstances. You know, so yeah. I guess I would definitely do that.
So if I may have follow-up comment.
Yeah.
My own experiences with TAs is you're the head coach.
Yeah.
And the TAs are assistant coaches, therefore must implement the head coach's vision. And that's because when you have two totally different approaches on the TAs, it creates actually different type of chaos that is undesirable.
Yeah.
And from my own experience, I figure out what you want and have the assistant coaches implement it in their own way.
Right.
So they have ownership of it, but if it's too,
that's
if you have competing things coming in, it creates a problem. And both can be great. There's nothing, it's not that one is right and one is wrong. That's not the issue. The issue is one, they need to be your vision.
Sure, no, and it's very much my vision right now, I would say, but also the implementation is, you know, one TA likes to really walk through all of the steps ahead of time, and we don't always have, you know, so sometimes I'm like, all right, I think we gotta, like, let's just start pipetting, and then we have so much support in this room that, like, we're gonna help people who have questions, like, I think there's a point at which you've talked too much and you've lost your audience, and that's a delicate line to find for an individual TA. You know, how much do I say? How much do I go walk through everything, and how much do I just let them go? And for some activities, it's better to talk more, and for some activities, it's better to talk less, and we're still kind of learning what those are.
My experience is less is always.
Yeah.
better. One technique that
Elizabeth says,
implemented was to
Elizabeth says all great changes are preceded by chaos.
Yes, yes, that's.
Yeah, so any other questions? Any questions from the Zoom audience? Okay.
It's fun.
All right, thank you,
Thank you.
all three of you so much, Amy, Jess, Madison. Thank you everyone online for joining us. They're
Let's give them a hand.
Okay.
Yeah, so today we're using.
So I will circulate the recording of this.
Oh, yeah, you have to turn it off on that one.
Yeah, I'll turn it up.
You know, so they're like Madison's usually like, okay, chaos, let's just go, let's do it. But then we're like, oh, now it's too much. It's like, that's some chaos. Yeah. Right. So today the other TA's approach really paid off because he was like, this is what